Hello everyone, we are the third year medical technology students from Notre Dame of the Jangas University. In today's video, we will discuss about a clinical study under the pathogen Enterobacteriaceae.
We are the group 5C, and the pathogen that we chose is Escherechia vulneris. And my name is Francis Gabriel Kibuko.
And my name is Romar Chu.
My name is Lloyd Vinces Pibasita.
And I am Renaline R. Halangos.
First, we will be having a short description about Escherechia vulneris. First, it is a rod-like bacilli, and it is under the genus of Escherechia. It is also facultatively anaerobic and non-spore forming. Facultative anaerobic means that with the presence of oxygen, it will be able to produce ATP through anaerobic respiration. And without oxygen, it can ultimately change into fermentation. Lastly, it can colonize in the respiratory tract, genital tract, stool, and urinary tract. In this video, we will be first having an overview, diagnosis of the patients, laboratory findings, figures, and tables. This case study was done by Tafik et al. And it is titled as Molecular Investigation of an Unusual Bacteria Causing Severe UTI Among Public Water Closet Users, Mechanisms of Characterization and Engineering Solutions. In our video, we will not be dealing with its engineering solutions and other molecular investigation, but we will be discussing about the laboratory findings focused on microbiology or its colonies and etc. So water closet is just a fancy word for bathroom. Next overview.
So, for this overview of this study, it states that a total of 100 employees in the age range of 25 to 60 years old from both sexes participated in this screen for UTI during period of September 2019 till February 2020. Second, they all work at Building R Educational Facility, and 80 of them use the unisex water closets during their work daytime, 2 to 3 times a day. And lastly, the rest of them never used the workplace sanitary facility at all and were considered as controls. Males and females ratio 1:1 in each study group.
The evaluation of UTI prevalence among employees. For data collection, they use questionnaire. Urinalysis test is clean catch midstream fresh urine samples of 30 ml collected randomly. The clean catch method is used to avoid contaminating the urine sample with the bacteria that are normally present in the urethra and appear in avoided urine samples. It is used for routine urinalysis, urine culture, or other urine tests that require uncontaminated urine for accurate results. In urine evaluation, reagent test strips is used. So the following test paddles are commonly featured on the reagent strips, which is the blood, bilirubin, urobilinogen, nitrite, leukocyte, protein, ketones, glucose, pH, and specific gravity, which is later on we will be discussed in the other slides for the result. And then microscopic examination is the 10 millimeters of each urine sample centrifuge for 5 minutes at 3000 RPM.
Next is the cultivation of the infecting microorganisms, and in this part, there are different types of culture media used in the study. Includes the Nutri agar, Limki agar, and blood agar from high media and India. So first, gram staining. So as we all know, gram staining is a bacteriological laboratory technique used to differentiate bacterial species into two large groups, which are the gram positive and the gram negative. Next is the analytical profile index system. So this system is used or developed for identification of clinically relevant bacteria. So in this case, the study used the API-20E kit, which is specific in identifying gram-negative bacterial family Enterobacteriaceae. Lastly is the Kirby-Bowers diffusion method. The purpose of Kirby-Bauer diffusion method is to determine the sensitivity or resistance of pathogenic aerobic and facultative anaerobic bacteria to various antimicrobial compounds. So in this case, Weller-Hinton agar or MHA is used with 1 ml of bacterial suspension.
Next, we have the diagnosis and the laboratory findings.
So, the total of 100 employees subjected to extensive question within the study questionnaire, and the result, consequently, the result of Table 1 encouraged the assumption of the incidence of urinary tract infection among the employees who use the water closet at their workplace, the building R. Next.
For Table 2, these are the results of the reagent strip parameters. As what you can see in the table, leukocyte and nitrite have the highest have result, which is the 100%. So, in screening the results of urinalysis test, reagent strip method of urine samples collected from 80 participants suspected with urinary tract infection enrolled in the study. So, the presence of nitrites in the urine is highly specific to a certain bacterial infections. Nitrites do not occur with all types of bacteria, and leukocyte suggests that there are white blood cells in the urine that may cause urinary tract infection. The presence of a positive nitrites in the urine indicates the presence of an organism that reduces nitrate. However, not all urinary pathogens are nitrate reducer. A positive test is highly specific for bacterial infection, but a negative test does not exclude infection, giving this test a low sensitivity. So, if a positive test for nitrate, it is called nitrituria. If you have nitrituria, your doctor will likely want to send your urine sample to a laboratory for urine culture test. In a urine culture test, your doctor can find out which specific bacteria is present that are causing UTI.
So next, we have the table 3. The table 3 shows the types of gram-negative bacterial isolates for per urine sample and also the types of grams-positive bacterial isolates per urine sample. So in this microscopical examination of isolated bacterial colonies, revealed 2 gram-positive isolates of Staphylococcus aureus and Staphylococcus epididymis, with the predominance of a novel grams-negative rods of Escherechia vulneris. In this table, it shows that it is clearly potent that this is not a normal UTI because we can see a high value of Escherechia vulneris and 65% of the sample is positive for Escherechia vulneris. And the second leading bacteria is the Escherichia coli, which is the common pathogen for UTI. Next.
So next is the table 4. So in this table shows the classification of UTI patients enrolled in the study according to clinical presentation and severity level of infection displaying cystitis, pyelonephritis, and urosepsis. So let us first define what is cystitis. So cystitis is inflammation of the bladder, and most of the time the inflammation is caused by a bacterial infection, and it's called a urinary tract infection. Next is the pyelonephritis. It is an inflammation of the kidney, typically due to a bacterial infection. So symptoms most often include fever and flank tenderness, and other symptoms may include nausea, burning with urination, and frequent urination. Lastly is the urosepsis. It is a term used to describe a type of sepsis that is caused by an infection in the urinary tract. It is a complication often caused by urinary tract infections that are not treated quickly or properly. So in this table, it shows the types of UTI employees wherein In cystitis, there is 74.79 in female and 71.2 in male. Then in pyelonephritis, there are 22.71% in female and 21.3 in males. Then lastly, in urosepsis, there are 2.5% for female and 7.5% for male.
As what I have said in the previous slides that if your result for nitrites and leukocyte is positive, your doctor were more likely to request a urine culture. A urine culture usually takes about 2 to 3 days to be completed, sometimes longer depending on the type of bacteria. On average though, you could see the results in 3 days. And keep in mind that not all bacteria are capable of converting nitrite to nitrite. So here in the figure, you can see E. vulneris, E. coli, and K. pneumoniae and staph epidermis, which are the results from the culture. The positive leukocyte and nitrite urine samples with a bacterial count over 105 UFC or ml were cultivated according to the standard procedure on different identification culture media. And these are some photos of positive culture growing on makanki and nutrient agar.
So for the figure 2, it shows that the fermentative E. vulneris has grown on Mach-1K agar at 37 degrees Celsius, and colonies, as presented in the photos, of look generally smooth, creamy with shiny surfaces and low convex. Besides, gram staining of isolated colonies revealed gram-negative, non-spore forming motile rod-like or bacilli.
Next, we have the antibiotic stability testing. The result of the researchers showed that all strains were uniformly susceptible to cephalosporins and ciprofloxacin. In contrast, most of the vulnerary strains were resistant against these following um, antibiotics. For the conclusion, first we have employees revealed cystitis, pyelonephritis, and urosepsis associated with the Escherichia vulneris as the main etiological factor of the infection. Next, we have isolated strains of Escherichia vulneris revealed a broad spectrum of antibiotic resistance and susceptibility to only a few. So kanina, ang susceptible lang siya doon sa dalawang antibiotic. Next, we have unisex water closets at workplaces with low performance values and responsible sanitary hygiene behavior impose an extra burden on the health of the employees as it might promote UTI. To our knowledge, this study or this thesis is the first defined case of E. vulneris associated with the community-acquired UTIs among people sharing the same water closets. So, naglabas po sa conclusion na yung company na ginatuluyan ng mga employees or ginatrabaho na employees, they don't practice sanitary measures sa kanilang mga bathroom. And that concludes our presentation. Thank you for listening.